Welcome to Genialis Expressions. Today I'm going to briefly walk you through the Genialis Expressions app to show you how you can analyze, manage, and interpret your RNA-seq data without having to rely on bioinformatics scripts or collaborators. Let's walk through a fairly common experiment. We're going to analyze the RNA-seq data from a study on the effects of the JQ1 small molecule inhibitor on triple-negative breast cancer cell lines. We have 4 experimental conditions: drug-resistant cells treated with either DMSO control or the JQ1 drug, and non-resistant cells treated with DMSO control or JQ1. Each treatment group contains 2 replicates, so we have 8 samples in total. Here you can view the details about the automated pipeline that was used to analyze the sample in the primary analysis history card. And here you can annotate your sample with information about the experiment such as organism, source, and cell line. We'll see how these annotations can be important for interpreting your data in a moment. Let's begin on the Sample Comparison tab by asking, are my replicates consistent, and are my experimental treatments and controls distinct from one another? In the hierarchical clustering plot, we see that the replicates form clades as expected. We can also see that the drug-treated non-resistant cells cluster quite distinctly from the control groups. We can see similarly paired clusters in the PCA plot, thus validating the experimental design. Next, we can ask, do my known marker genes display expected expression patterns? Since JQ1 is a small molecule inhibitor in the BET pathway, we can further validate our experiment by determining what effect JQ1 treatment has on genes in that pathway. For this, I've selected GPI, which is a housekeeping gene and whose expression should remain unchanged. I've also selected MYC and CCND1, whose expression is regulated by BET proteins and we expect to be downregulated. Lastly, I've selected HEXIM1, which is known to be upregulated by BET inhibitors. And when we look at our bar chart, we see just that. Compared to the expression of the housekeeping gene, GPI, both MYC and CCND1 are downregulated in drug-treated cells. HEXIM1, known to be upregulated by BET inhibitors, is similarly upregulated in our experiment. You may have noticed that 2 of the samples are colored differently. As I'd noted earlier, we'd previously annotated those samples according to their treatment type, which allows us to easily distinguish the treatment groups during our analysis. The next thing we might want to ask is which genes are up or downregulated in response to drug treatment. In this example, we can see that there are 176 genes that are upregulated and 327 genes that are downregulated in response to JQ1 drug treatment. Since we're primarily interested in understanding which cancer-related genes are downregulated, we can highlight those genes in the volcano plot to investigate them further. Once we've selected our downregulated genes, we can visualize just how different their expression is across each experimental condition in a heat map. In the heat map, we can see the entire list of downregulated genes. Here we can also see that CXCL1 expression is significantly downregulated in non-resistant cells treated with JQ1. Let's say we're not familiar with CXCL1. We can simply search for it in the genes card, and then we can scroll down to view its description from NCBI. Here we can see that this gene is involved in the growth and progression of various cancers. Now we have a potential drug target that we can investigate further. Finally, you can download your data in publication-ready images. See firsthand how Genialis can take you from raw RNA-seq data to actionable insights in a matter of minutes. Request your free trial at www.genialis.com.